We will now look at chemical digestion in the small intestine. So first, what is the pH in the small intestine? Is it acidic, neutral, or basic? The answer is basic. Great. So let's try to see the digestion of meat in the small intestine. Let's bring back the key. What do you think will happen if we add lipase? Let's see. Lipase could be added because lipase works on basic pH. Lipase is going to digest the fat and not the protein. It happens in 3 steps, and the fat is digested in the small intestine. Now let's add protease to digest the proteins. You can see that the proteins are digested by protease, and the fats, of course, are not. Notice that the proteins here are broken down into their individual units. They're broken down into amino acids. Meanwhile, in the stomach, when proteins were digested, they were not broken down completely into amino acids, they were only broken down into peptides. So this is a key difference between the digestion of proteins in the stomach and in the small intestine. And notice that carbohydrates can be added to the medium. However, what is it going to work on? Carbohydrates cannot digest fats or proteins, so it doesn't really do anything for the digestion of meat in the small intestine. So this was meat in the small intestine. Now let's look at bread. We add lipase. We know that lipase can work in the small intestine. What is it going to digest? Which one do you think will be digested? Exactly, it is the fats, digested in 3 steps by lipase. Now we add protease. What do you think it's going to digest? It digests only the proteins, and it completely digests the proteins. It breaks them down into their individual amino acid units. Carbohydrates is added, and what is it going to digest? It is going to digest the carbohydrates, and this time it's going to digest them fully into their individual monosaccharide units. Unlike in the mouth, where carbohydrates were not fully digested into monosaccharides, they were broken down, for example, into disaccharides. So this is the digestion of bread in the small intestine. Now let's add butter. Let's see lipase. Lipase is going to fully digest lipids in the small intestine. We already saw this with meat and with bread. Of course, we can add protease because it works in this medium. However, there is no proteins in butter to be digested by protease. The enzyme protease does not work on lipids. Same thing with carbohydrates. It can be added to the medium, but it can't work on lipids. So it's not going to digest the fats. So this was digestion in the small intestine. We have the complete breakdown of all food groups into their individual units.